Hi, dear students, hope you are doing well. In this video, we shall be studying the introduction to enzymes, their structure, and their mechanism of action. Let's get started. Enzymes are biologically active proteins that speed up biochemical reactions, making life possible. An enzyme contains an active site, a globular protein structure, and a cofactor. Active site is a small region where the substrate binds, enabling catalysis. Globular structure is the overall shape of the enzyme, composed of hundreds of amino acids. Cofactors are non-protein components, such as metal ions or coenzymes, that aid in catalysis. The cofactor usually acts as bridge between the enzyme and its substrate. Often it contributes directly to the chemical reactions which bring about catalysis. Sometimes the cofactor provides a source of chemical energy, helping to drive reactions which would otherwise be difficult or impossible. Some enzymes use metal ions as cofactors like Mg2+, Fe2+, Cu2+, Cn2+, etc. The detachable cofactor is known as an activator if it is an inorganic ion. If the non-protein part is covalently bonded, it is known as a prosthetic group. If it is loosely attached to the protein part, it is known as coenzyme. Coenzyme is closely related to vitamins, which represent the essential raw materials from which coenzymes are made. Only small quantities of vitamins are needed because, like enzymes, coenzyme can be used again and again. An enzyme with its coenzyme or prosthetic group removed is designated as a aponzyme. Adding the correct concentrated coenzyme to the aponzyme will restore enzyme activity. An activated enzyme consisting of polypeptide chain and a cofactor, known as enzyme. Many enzymes are simply dissolved in the cytoplasm. Other enzymes are tightly bound to certain subcellular organelles. They are produced by living cells for use in or near the site of their production. The enzymes important in photosynthesis are found in the chloroplasts. And enzymes involved in cellular respiration are found in the mitochondria. Some of the enzymes which are involved in the synthesis of proteins are integral part of ribosomes. Mechanism of enzyme action. Enzymes recognize and react with specific substrates, transforming them into products. Enzymes act in a series of chemical reactions, often in a specific order, to complete metabolic pathways. Enzymes are very specific in their action. A single enzyme catalyzes only a single chemical reaction or a group of related reactions. Sometimes enzyme action may occur in an enzyme-to-enzyme chain, during which substrate molecules can be literally handed on from one enzyme to another to be catalyzed furthermore. Now, we see how does enzyme-substrate interaction take place during a catalysis. The active site of the enzyme is responsible for binding to the substrate. The active site consists of two regions, the binding site and the catalytic site. The binding site recognizes and binds to the substrate, activating the catalytic site. The catalytic site then transforms the substrate into products. Enzyme action models. Two models describe enzyme-substrate interaction: lock and key model and induce fit model. Lock and key model was proposed by Emil Fischer. This model suggests that the active site of an enzyme is a rigid structure that only binds to a specific substrate. There is no modification or flexibility in the active site before, during, or after the enzyme action, and it is used only as a template. Therefore, later studies did not support this model in all reactions. Induced fit model was suggested by Koshland, which suggests that the enzyme structure changes when it binds to the substrate. The change in structure enables the enzyme to perform its catalytic activity more effectively. An enzyme and its substrate react with each other through a deionite charge-bearing site of an enzyme called active site. The charge and shape of the active site is formed by some amino acids present in the polypeptide chain of the active site of the enzyme. These amino acids are brought closer and are arranged in a specific way by coiling and folding of the polypeptide chain within the globular symmetry of the enzyme. The active site of the enzyme is made up of two definite regions. That is the binding site and the catalytic site. The binding site helps the enzyme in the recognition and binding of a proper substrate to produce an enzyme-substrate complex. This reaction activates the catalytic site. Activated catalytic site catalyzes the transformation of the substrate into product S. Thus the enzyme, after catalysis, detaches itself from the products unchanged. Enzyme requires aqueous medium for its activity. Most enzymes do not float about in a kind of cytoplasmic soup, but are attached to membrane systems inside the cell in specific and orderly arrangements. Mitochondria and chloroplasts are good examples of this.